Okay, folks, we're going to get started now. As people walk in, they'll get introduced to the subject matter, but I think we've got a quorum. Today, my name is Angel Pizarro. I lead the global initiatives for genomics and life sciences for Amazon Web Services as part of the scientific computing team. It's going to be my pleasure to announce later Dr. Ben Langmead from Johns Hopkins University is going to be talking about some applications they've built on top of Elastic MapReduce service, as well as Abid Nalor, who is now at OHSU, or soon to be at OHSU, yeah, Abid. And you guys will be discussing RailRNA. But for first, let's take genomics at scale. Using AWS for population scale analysis of genomics and life science data. The agenda for today is I'm going to give an overview of Elastic MapReduce, which some of these systems have been built on. We're going to do a review of RailRNA, again, the algorithm that Johns Hopkins University built on top of Elastic MapReduce and some other other infrastructures as well. And if we have time, we'll look at more EMR systems for science and why you want to use scalable systems like Elastic MapReduce for science problems. We will preference a question and answer session above that last section. So let's see where we are in terms of time. Let me start off my stopwatch there. So what are the challenges with in-house infrastructure? One, it's a fixed cost. Oftentimes when you're looking at analyzing for data at scale, you're looking at capital expenditures, and that fixed cost will need to be amortized over 5 years, or really 3 years, but academia, you take it 5 years. Your storage and compute are co-located. It's always on. It's not scalable, so oftentimes you'll have an infrastructure that is at a certain capacity where you have a maximal problem size that you can solve with it. Outages, I don't know how often or how good your operations is, but the outages happen to everybody. And for a single resource within the campus, the outage can impact a very broad community. Another in-house infrastructure challenge is production upgrades. What does that mean? Well, these systems are in use almost 24/7. In order to expand or build upon the system, you actually have to do it on the production system often. And there's slow deployment cycles. So how often can you bring in more capacity, more compute, more storage? But really, especially for Hadoop systems, this came out of an ecosystem where compute and storage grew together. They're built out of systems that you just take, white-labeled systems that had a bunch of disks, and you tie these nodes together into a resilient and scalable platform. But if you wanted to grow the system, oftentimes you had to grow the storage along with the compute. Oftentimes, especially in genomics, your compute requirements vary widely from your storage requirements. And here's an example of it, right? So imagine that the 100% line, not the 120%, but the 100% line here represents your overall capacity. And for the most part, you have weekly processing that might happen within this cluster as you're doing batch genome sequencing. At the end of that, you're going to have a set of data that's going to be batch processed. You might spike up for these weekly peaks, and then you might have some cohort analysis where you have to reprocess the entire data set, taking up the complete capacity of your cluster once or twice a year. This actually happened to me, right? I mean, when I was at University of Pennsylvania doing proteomics-based research, we had our own group cluster. And when it was time to replace that cluster, we took a look at the utilization graph, and this would have been fantastic for our utilization graph, quite frankly. We hovered at 10 to 20% utilization for most of the year, except right before ASMS and another proteomics conference where we spiked to 100% roughly twice a year, every year for 4 years. So we decided that with AWS, you can actually bootstrap the compute that you need at a point in time basis. And any of this gray area here, it is gray, trust me, any of this gray area here that's unused capacity, we weren't paying for it. We weren't paying for the electricity. We weren't paying for everything. But with on-prem infrastructure, you are paying for that maximal capacity. Right. So that's the value proposition of something like EMR. It's a managed, so what is it? It's a managed Hadoop platform. It has pre-installed MapReduce tools. These are the open source tools, Apache Spark, Presto, and a few other general purpose Hadoop infrastructure tools pre-installed and ready to go. You can launch a cluster within minutes. You have an open source distribution, so regular Apache. You also have the option to use commercially supported MapR distribution on top of VMR. And most importantly, you're able to leverage the elasticity of the cloud and baked in security features that we offer with respect to utilizing encrypted data stores in transit and in flight, as well as some simple configuration options that will allow you to secure your infrastructure very easily. Finally, you can pay by the hour, right? So if you only need to process something for 3 hours a night or once a week for a couple of days because your genome sequencing came off of the instrument at that time, or your image analysis and is ready to go, you can choose to bootstrap your cluster just for the time that you use it. And in particular, we have this thing called the spot market, which, the spot market, which I believe Ben will be talking about a little bit, that allows you to save even more money. So, easy to use, low cost, elastic, reliable, secure, flexible. That's the value proposition of Elastic, MapReduce, or any of our managed services. They grow when you need them to, and they shrink when you don't. In particular, one of the bigger strengths with EMR is the fact that you can decouple the storage from the compute. Because these things are elastic, because you can define, I need this many compute cores, but I only need to work on 10 terabytes of data. That means that you can put in thousands of compute cores accessing the same 10 terabytes of data, whereas the on-prem infrastructure, you might need to put in X amounts of storage every time you submit a node. So if you need a 3,000 core cluster, usually for Hadoop, that means that you put in quite a large storage infrastructure that goes along with it in order to make the economics of that work. In particular, we use Amazon S3 as our persistent data store. Within EMR, there's some reasons you want to use Amazon S3. First is the price. It's 3 cents per gigabyte per month. This is based on the US East region prices. There's 11 nines of durability. That means that it's very resilient data. There's 3 copies of your data the minute that you put it in there. We do continuous checksumming of data as we move it around the system. There's rich lifecycle policies that you can use to say, I want this data that's been up here for 3 months to be put into an archive tier that's a lot less cost because I know I haven't accessed it beyond that. There's versioning, it's distributed by default, and in particular, it forms the basis of something we call EMRFS, or EMR file system. So why is S3 good for genomics? Basically, since you're paying per gigabyte cost, you're actually paying for the storage that you're actually using at that time. So it's not appropriate to do a cost estimate. Well, how much does 100 gigs of S3 cost me versus a 100 gig disk? That's the wrong question. The right question is, how much data do I have over time, and what happens when I provision that data over time in terms of my cost? You pay exactly for what you use. You can have object sizes up to 5 terabytes. I was talking with somebody the other day that received a 30 no, it wasn't 30 terabytes. It was a 30 gigabyte VCF file. I mean, that's a giant set of genome information. They couldn't work with it in Excel. They couldn't work with it in a lot of different frameworks. They needed to transform it into something else. It's very high bandwidth. It's a horizontal bandwidth. It's durable. It's fine-grained access controls, and a lot of other reasons you want to use S3 for genomic data. But in particular, you want to use S3 within EMR because of EMRFS. EMRFS allows you to leverage S3 as a file system. It streams data directly from Amazon S3. It uses HDFS intermediates in order to cache data at the proper time. And it has better read-write performance and error handling than other open source components, right? So Within this, it's assumed that EMRFS is specific to EMR, right? It's right there in the name. There are open source alternatives that do use S3 as a backing HDFS file system. We've done a lot of work with the EMRFS to make sure it works with our infrastructure resiliently and performantly. It has a consistent view, which means that when you read something, if you want to write, you can define that this thing will be there when I do request it. And it has support for encryption and fast listing of objects. So what does it mean to actually use EMRFS in practice? So here's I believe this is a Hive query. At the bottom here, you'll notice it's a location of some HDFS endpoint, which is samples, pig, Apache input. You want to create an external table from this data source. With EMRFS, that's the change you need to make, which is really pretty powerful, right? So now you have all of your data within S3. You don't have the HDFS backing store that you normally would have had, and you've successfully decoupled your storage from your compute. There's still some amount of HDFS storage that we recommend as scratch space to read in data in and out and do random read operations, but for the most part, a lot of your data isn't being utilized all at the same time. You can create that HDFS resilient storage to meet your exact demands for the job. So with EMRFS and EMR, you're able to build transient clusters that you can bootstrap up and down, and you can take that same data and shift it into other solutions, whether these are traditional HPC clusters that are running on top of S3, other EMR clusters that are using the same data going into our Redshift Open Data Warehouse, and whatnot, as well as other types of algorithms like machine learning, running R, C3, C-type instances. Right, so at this point, that's the background you need to understand what the motivation was to adopt EMR for building science tools, and I'd like to bring up to the stage Ben Langley.
Great, thank you. So my name is Ben Langmead. I'm an assistant professor of computer science at Johns Hopkins. I've been using Amazon Web Services for quite some time, since around 2008. I'll say a few words about what it is we do in my lab, why Amazon Web Services is a good fit, and some broad strokes information about what Amazon services we use and for what, and then I'm going to hand it off to Abi Nalor, who knows a lot more than I do about the particulars of how we use it, AWS. So if you read the newspaper, you're the popular press, you're probably aware of some recent huge breakthroughs in DNA sequencing. So DNA sequencing is something that we sort of take for granted these days, but as recently as the Human Genome Project, it was something that not a lot of people really knew a lot about. These days, DNA sequencers are incredibly fast, they're incredibly cheap. The sort of number of bases of A's, G's, and T's that you can read per dollar using a sequencing machine has been going up very, very quickly, faster than Moore's law. The sequencers themselves have got smaller and more convenient to use. They're about the size of a mini fridge. You can fit a bunch of them in a laboratory. And with these advances, lots more people have been using them. They've become kind of the go-to tool for analyzing any phenomenon that has to do with nucleic acids like DNA or RNA, and we'll mostly actually be talking about RNA today. And so with DNA sequencers making their way into so many labs, it's become kind of a leveler. It's become much easier for people to obtain large amounts of life science data. Even small laboratories, laboratories that look a little bit less like this laboratory, which is a big one, a little bit more like this, which is a not so big laboratory. So I come from Johns Hopkins. We're sort of a loose confederation of small laboratories. We're not really a single big lab kind of a place. So while these advances in technology have sort of created the potential for everybody to share in this sequencing revolution, one of the biggest barriers is computation, just the availability of appropriate computational resources and computational savvy, because not every biology lab is really able to employ the sorts of people who would be needed to take advantage of any kind of computational resources, let alone cloud resources. So one of the things that we try to do is to try to make it very, very, very easy for life scientists who aren't necessarily very computationally well-resourced or savvy to make use of sequencing data. And in particular, so if the invention of these new DNA sequencers is kind of like the printing press or something like that, all of this data is going into a big library. So there's an archive in Bethesda at the National Institutes of Health at the NCBI called the Sequence Read Archive, which is where a lot of researchers who publish genomic studies are sort of compelled to upload all of their raw data so that it's available to other people. This is all taxpayer funded, so we want it to be available. We want other people to be able to reproduce those results or come to new conclusions using that existing data. So we now have this huge resource. It's many petabases, so peta ACs, Gs, and Ts worth of data stored in this resource. And this is very valuable data. So even though sequencing itself is, you could say it's cheap, some of this data comes at a very high sort of opportunity cost. So some of it is derived, for example, from people who have rare diseases, or it's some sort of hard to obtain tissue. Some of it is multiple tissues from the same individual. These are just not the sorts of data sets that most people can get their hands on, so a lot of it is very valuable. And it's publicly available. Some of it, so this is pretty hard to read, but this vertical scale here is basically the number of bases in the archive, but it's on a log scale, and the horizontal axis here is just calendar year. And one thing that you can't really tell from the graph, but just in the last 18 months or so, it has doubled. So the doubling time for this archive is about a year and a half. And there's two lines here. There's a very light yellow line that you probably can't see, and then there's a blue line. And the difference between these two lines these days is about twofold. So this is about a twofold difference. You can't tell because it's a log scale. And the yellow line is the number of bases that are open access. Anybody can download them today. And the other one is bases, total bases, which includes protected bases. So we're going to talk a little bit today about how you analyze data that's protected because it's derived from living human patients for whom the data is, you're only consented to work with the data in certain ways. So that data is actually protected by certain security measures. But we'll talk today a little bit about how you can still analyze it in AWS even with those security measures. So I've been working on trying to analyze data like this in Amazon Web Services, like I said before, since about 2008. So that was before, I think it was before the advent of Elastic MapReduce. I certainly wasn't using it back then. Back then, firing up an EC2 cluster was, there was sort of like a cumbersome pile of bash scripts, and that's how you did it. Then Elastic MapReduce came along, and that's when things, I think, from a biological user's perspective, really started to get good, because it meant that you could potentially start to fire off huge analyses of genomic data using just one or two commands on the command line. And a little bit later, Abhi will show you that you really can do 2 commands and analyze a huge genomic data set on Elastic MapReduce. It's kind of like the command line, as far as we're concerned, for using Amazon Web Services. We use several services from AWS, including EC2 and Spot, as I just mentioned, or EC2 and EMR. But also, we use the Spot marketplace. As Angel mentioned, this is a good way to get lots of computational resources on AWS for cheap, because on the Spot marketplace, you're bidding on excess capacity, and so the price is a fluctuating market price that usually is substantially lower than the on-demand price, something like maybe 5 to 8-fold lower or something like that. So you can get your hands on lots of capacity all at once, as long as you don't mind the potentiality that if the market price goes above your bid, you might lose those resources partway through your computation. But often that's something that you can tolerate, and in this application, it's something we can tolerate. So we use Spot extensively, and we use Amazon VPC and CloudFormation as part of our architecture for being able to analyze protected data. The privacy standard is called DB Gap. So in order to implement our DB Gap compliant data analysis, we use these 2 services, as Abhi will talk about a little bit more later. This is part of a sort of history of work that we've done in our lab for analyzing genomics data on AWS, stretching all the way back to this paper that we published in 2009. That was not using Elastic MapReduce. But then these 3 papers, which all have to do with analysis of RNA sequencing data, those all use Elastic MapReduce. Our most recent effort, which Abhi will talk about today, is the last one listed here, RailRNA. so I think that might be, yeah. So with that, let me hand it over to Abi Nalor. He's going to talk more about the particulars of RailRNA and how it uses AWS services. Forward, backward.
Great. Okay, yeah, so I'm Abi Nalor, and I'm currently a postdoc at Hopkins between Ben's lab and also Jeff Leake's lab. And yeah, I will be at OHSU in the fall as an assistant professor. I'm going to be telling you a bit about the technical details of RailRNA. When we started developing RailRNA about two years ago, the most widely used splice alignment software was TopHat2. It probably still is. And TopHat2 was designed to run on a single sample at a time. It is composed of a few steps that are pretty long and don't take full advantage of available compute and run on a single core. And that's fine for its purposes, but we really wanted software that scaled to analyze many thousands of samples. So we fit the spliced alignment problem, which is what Rails solves. It analyzes lots of RNA sequencing samples, which necessitates spliced alignment. We fit this spliced alignment problem into the MapReduce framework as implemented in Hadoop. And this allows us to analyze thousands of samples on tens of thousands of processing cores in a way that's fault tolerant and reproducible. And it's especially reproducible if you use elastic MapReduce, because you're running the same software on the same standardized units of hardware, the same on EC2 instances. Hadoop also allows us to aggregate information, easily aggregate information collected from across many samples. So for instance, we can juxtapose in our outputs the samples in which we find the same feature. So for example, let's say we have some threshold for genome coverage above which we say a gene is expressed in a sample. What we can do is make a coverage, an interval-based query on a sparse junction by sample matrix, very rapidly obtain the samples in which a gene is expressed, simply because of the way the data is arranged. And that's made possible by, that's made very efficient by using Hadoop. So this is just a depiction of several steps of the pipeline. We wrap bowtie, which is alignment software that was developed by Ben. And we engineered Rail, so it's really easy to use, like Ben mentioned. We said in 1 to 2 commands, you go from a list of the URLs that point to the raw data to results. The results will be stored on S3. So here in this command, you'll see that there's a manifest file that's listing the URLs of the raw data. You specify a species to which you align your data. This is H338, so it's an assembly of the human genome in this case. You specify an output bucket on S3, and you also specify the size of your EC2 cluster, your elastic MapReduce cluster, and that's the core instance count, and the kinds of instances, the instance types that are in your cluster. So, it's as easy as 1 to S3. Yeah, that's the only joke in my part of the talk. It's not even really funny. Okay, so here, this is just an example of a set of job flows that we are running. And you can just see, there should be $ signs all over this when you look at this, because there's so many different job flows that are running here. This is actually from an analysis of 50,000 publicly available human RNA sequencing samples that are available on the Sequence Read Archive. We performed it in February. And this is all available human RNA-seq samples. So yeah, 50,000, it's about 100 terabases. We have a paper on this. It's called Human Splicing Diversity Across the Sequence Read Archive. You can read it on bioRxiv. And we did all of our analysis for this on elastic MapReduce. It's repeatable. You just visit this GitHub repo, and you can see the commands that will reproduce our results, same software on the same hardware. It's also relatively inexpensive job for us, coming in at about $1.40 per sample. And if you compare to sequencing costs, I think it can cost like between $20 and $40 per sample for sequencing. So compared to that, the analysis portion, if you perform it in the cloud, is pretty inexpensive, an order of magnitude less expensive. It's also a very rapid job. We went from input for results in about 2 weeks, just constantly submitting these job flows. We successfully submitted them. And the reason it took 2 weeks and not, say, a day, because remember, we can reserve as much compute as we want on EMR, is that you can only download the raw data so fast from NCBI. And so this was like our rate limiting factor in running our jobs right now. But for example, if all the data were hosted on S3, we could finish these job flows in a day. And that's actually now happening with the Cancer Genome Atlas. With TCGA, through a partnership with 7 Bridges, TCGA RNA sequencing data is now hosted on S3. And through the 7 Bridges API, you can now access those samples, the raw data for those samples on S3, and get temporary URLs to access them. We're actually building support for the 7 Bridges API into RailRNA right now, and hopefully in just a few months, well, probably just a couple of months, we'll be able to make some impactful statements about the speed with which you can reanalyze publicly available data that's hosted on a service like S3. Hopefully, making statements like this will encourage adoption of commercial cloud providers, especially Amazon, for hosting raw data and process data. So speaking of TCGA is DB Gap protected, which means that the NIH mandates certain security requirements for analyzing it. It's controlled access data, and it's controlled access to protect the privacy of research subjects. This is especially important for sensitive phenotypes like disease, which can be associated with a subject's identity. So we built support for analyzing DB Gap protected data in the cloud with Amazon Elastic MapReduce into RailRNA, and you can do it yourself. You pick your favorite DB Gap protected data set, and you can visit docs.rail.bio/dbgap to figure out how to get up and running with RailRNA to analyze the data. The way we do this, and the way this worked, was that NIH started allowing people, I think almost 2 years ago now, to analyze DB Gap protected data in the cloud as long as they released this paper that had certain requirements. And Amazon followed up with a white paper co-authored by Angel, actually, that specifies the particular Amazon Web Services that allow you to adhere to those requirements. So we took that white paper and we implemented it in Rail, and this is a protocol we call RailDB Gap. And we always launch our job flows into a virtual private cloud, a VPC. We ensure that data is encrypted at rest and in transit between the cluster, the EMR cluster, and S3. On S3, we turn on SSE, server-side encryption. In the cluster, we ensure that the ephemeral space is encrypted with LUKS, Linux Unified Key Setup. And between cluster and S3, we turn on SSL. So there you have it, end-to-end encryption. We also use security groups, stateful firewall, to block inbound access to the cluster by all services except EMR Essential Web Services. And we also use CloudTrail. We turn on CloudTrail to log account access and actions as required by NIH. And we make this easy for users to do. I mean, users, all this stuff here on the previous slide can remain opaque to users because we put it all in a CloudFormation template. And so a user need only create a stack from this template, and this stack will set up a secure S3 bucket with SSE turned on, set up the necessary security groups. The user just only needs to know what the name of the stack is, and then the user just specifies the stack name at the command line in that RailRNA command, submits the job flow, and doesn't really need to be aware of that they're compliant, but they are, which is great. So we're trying to make this product pretty usable so people can skip a lot of the technical details, and we hope we've done that. So yeah, again, docs.rail.bio/dbgap to get up and running with the DB Gap on EMR. And these are 3 papers that I touched on in my part of this talk. RailRNA, HTTP rail.bio. You can read our RailDB Gap paper, which was now published and accepted and published in Bioinformatics. And human splicing diversity across the sequence archive is our analysis of splicing across the 50,000 samples we analyzed in February. You can find a link to the paper at intropolis.rail.bio. Thanks a lot. I'm going to hand it back to Angel now.
Thanks, that was great, guys. We've got about 15 minutes left, so I think I'll just cover the last section of talk real quickly, because I think the motivation is an important one to get across. Oh, look at this. You guys forgot to acknowledge your lab. Oh, man. Oh, okay. I don't know most of these people, but this guy right here, don't trust this guy. Don't trust that guy. All right. Oh, yeah, that's true. We did support, so there is a credits for research program, by the way. If you guys have some idea that you want to try out on AWS in a low risk, low cost way, it's just a web form. You describe what you want to do. We have folks contact you and talk about it and say, okay, well, this is your research problem that you want to try and solve. We think that this architecture is probably the best one for it. We're more than happy to work with you. So just go to our website /grants and you'll see the credits for research program, which can help you also do your research and try these types of infrastructures out in a low risk way. So these guys do great work. Okay, so why is horizontal scale important? Why is it important for genomics, or why is it important at all? It's not just about chunking through data. I'll remind folks that this is essentially what happens with genomics, that you have some alignment steps, some deduplication step, and recalibration filtering, QC and filtering, variant calling, variant annotation, right? So this is sort of the golden path of human DNA sequence analysis. This is the DNA part, not the RNA part, but they follow a similar routine where they do alignment, calibration, and downstream analysis. This, in particular, is the GATK best practices. So the stages are written separately. There's a hand up, off. In between each one of these stages, there's usually some flat file that gets stream processed and fed into the next version. So you could actually do all of this within memory and Unix process pipes, but most people just write out the files. And everybody has their own flavor of this pipeline. So there's a need for integration. But parallelization, either in the cloud or HPC systems, tends to want to be like this, where you take one sample and process it through the nodes, or a server or an instance, and just have a completely disconnected pipeline. They didn't talk about it, but RailRNA does almost this. There's actually the pre-processing step, which was mentioned, is core to why RailRNA is a different sort of algorithm, in that the more samples you give RailRNA, the cheaper it becomes. Because that pre-processing step says that these sequences that are shared amongst them, even though we don't know where this goes in the genome, we know these sequences are the same. We don't have to align both of these things at once. So it's a quick shift in the way that you think once you start dating at scale, that the more data that you're producing, the more economies of scale that you get. And that's the paradigm shift that needs to happen across all of these big data sciences that in the life sciences, is that you need to start thinking about the problem in a different way that scales the per unit cost with the amount of units that you have, so that it goes down. All right? So for most of genomics, it is flat files, and in this particular format, this is a BAM formatted file, and you see that it's pretty much columnar text data, and it has some variable amount of unstructured information at the end, but for the most part, the large portion of it is structured information. But we're at a point where these file formats become our rate limiter. Right? Yes, you can take per lines and distribute it amongst the cluster, but these equate to file handles. And this is a portion of the Picard code. You're actually limited. You're actually limited within Picard, or at least one version of it, to 8,000 samples at once. And then when you're talking about population scale genomics, you might want to analyze more than 8,000 people, because there's complex disease phenotypes out there, and we need very large numbers, in the tens of thousands, if not the hundreds of thousands of individuals, to tease out whether some genetic phenotype is actually valid for the disease condition that you're talking about. So our community at large needs to shift away from arbitrary limits that are imposed by our architecture. And in particular, when we know very well that if you start doing this step, which is the variant calling, is this thing individual, this change in the individual stable across a population, and is it important for disease, you actually have to collapse down all of your parallelization into this single-threaded step. That sucks, right? You're now dealing with thousands of files within one system, and it's just not gonna work. It doesn't work. So, flat files are a blocker to population scale genomics. And in particular, there's lots of work within this field to look at Spark, which has a different computational model, a different semantics about how data is moved around to enable large-scale data processing. It's massively parallel, and it uses directed acyclic graphs to define the computational model as how you go between steps. And in particular, it's incredibly good at summations and iterative summations across different products. And I got to tell you, bioinformatics love probabilistic models. They're in all over the place. And if you have an architecture and a system that can deal with this mathematical concept well, you're ahead of the game. So a lot of these efforts are being led by and have come out of the AMP lab. There's a GitHub project under Big Data Genomics. You should check it out. Atom is pretty cool. It uses Spark, the Scala and PySpark to do a lot of primitives for genomic processing. And in particular, I'm just going to skip through so that we can get to a Q&A session. You can start loading VCF files into this Atom format, which is a gzip compressed Parquet file. And suddenly, you have the same data concept. So this could be, a source, tens of thousands of genomes within a VCF, split across a large distributed set of files that you can analyze in parallel across a horizontal scale cluster. So Atom's pretty cool. Check it out. Check out RailRNA. And I think, here's an example of dealing with genomic context of Atom files. But check out Atom, check out RailRNA, and I think we'll stop here and do some Q&A. So if Ben and Abi come on up. Any questions? Anyone? Step up to the microphone. No? Can you step to the microphone, because I can't, we'd like to have these for posterity.
Hi, good afternoon, and thank you
you.
very much for your chat. I was wondering if you guys could talk a little bit more about the regulatory and the legal environment that you work in. You described some of the security controls that you put inside your project to make you able to work on certain types of data. Could you describe a little bit about what compels you to do that? Do you have to accredit your systems in any way?
Yeah.
What does that environment look like for your use case?
So the genomic data sharing policy of the NIH actually stipulates that you meet certain NIST requirements for security and compliance. Things like encryption of data in flight, at rest, security firewalls that do not allow access from the internet. Basic IT security framework that you should have in place before you can transfer that data into that system for analysis. The RailDB Gap CloudFormation essentially bootstraps those security controls on top of AWS, following the guidelines of that genomic data sharing policy that the NIH does. We also have just released a Quick Start, which is an alternate CloudFormation template that does some of the same things. I still have yet to do a crosswalk between the two, so I'll have to look, but it covers NIST 171 compliance, and it also provides an Excel spreadsheet, which is the real value, that has that NIST control. What is the responsibility of AWS? What are our systems? And what is your responsibility as a customer? So if you take a look at an example, for instance, volume encryption. So you have a storage volume that you want to attach to an instance, and the security guidelines say that must be an encrypted volume. The control is you must have encrypted volumes. The AWS portion is we offer EBS, which allows you to encrypt volumes, both for data in transit and at rest. And then the customer responsibility is, yes, we've checked off that flag that says we are encrypting this volume, and it gets encrypted, right? So that security framework is really quite nice, and you should take a look. Just do a search for AWS NIST 171.
Hi, thanks for the presentation. I was curious for Ben and Avi. You guys are analyzing a lot of public data from NCBI, and curious to hear kind of what you're learning and what challenges you may be running across from data that are collected in different ways, have maybe disparate, inconsistent metadata. What sort of conclusions can you draw from lots of public data that you're trying to reanalyze, and how, I guess, robust or how confident are you in the conclusions that you can draw from those disparate public data sets?
I think that's a really good question, and you mentioned metadata, which is maybe one of the most important things I would hit on, which is we have all this data that's from a bunch of disparate studies, from a bunch of disparate laboratories. One of the biggest problems is that when they upload their data into this public archive, it should be annotated with enough information so that we can go back and figure out, for example, was this a human? That's a good thing to know. Were they male or female? What is the disease state? What laboratory did it come from? What exact protocol did they use in the lab in order to obtain the library that they actually sequenced? And the sequence read archive is an example of a place where they don't have a lot of very strict requirements for the people who are inputting that metadata, right? So the metadata is of varying quality. We know that there are a lot of mislabeled samples. We know that some samples are lacking the information we really need to make a good judgment about what to include in analysis. And that's a big issue. And so one of the ongoing interests in my lab and in my colleague Jeff Leake's lab that Avi also is working in is how do you, for example, check the quality of the metadata over a very large number of samples, and maybe even fix the metadata where it may be wrong or missing. And I have colleagues elsewhere that have also looked at this. Like, for example, when we analyze RNA sequencing data, we actually draw on third-party sources of metadata. There's a lab at Carnegie Mellon, Carl Kingsford's lab, where they've produced a huge collection of additional labels for a bunch of public RNA sequencing data sets, like cell type and tissue, that they obtained by not just taking the annotated metadata, but actually reaching into the abstract of the paper and doing some natural language processing to try to extract missing information. But the answer to the question, how do you do it, is very carefully, and that is definitely, it's very important that we tackle the problem of trying to make public data as usable as possible, because it's so valuable, and we the taxpayers are paying to keep it there. We absolutely have to use it. But part of that is going to be trying to do a good job of, in retrospect, trying to fix and otherwise understand the metadata that's there a lot better than we do now. Anything you wanted to add, Avi, about that?
Well, Ben covered some of. I was just going to add some technical issues with downloading lots of data. First, there are going to be samples that are incomplete, samples that just don't make sense. We can't interpret it. We hadn't anticipated that the data would take a particular form. So you have to you have to ensure that your pre-processing is robust to bad records, for example, records where, say, the read length doesn't match the quality string length. Generally, just using, for instance, SRA tools, fast QDump, it'll crash sometimes on some data sets. There will be inconsistencies. So those are the technical challenges with analyzing lots of data. There will always be some quirk of the input data that you haven't anticipated. That's part of the reason we actually divided our jobs across several. So you saw that screenshot where we had several different job flows. Part of the reason we did that is also that we know some of those job flows are just going to fail. If we put it all into one job flow and that job flow failed because of one bad sample, it would be bad news for our bank accounts. So, yeah.
Oh. Sure. We could repeat the question. Why don't you go and then we'll have that one. Go ahead. All right. Just say it. We'll repeat the question. Just say your question.
So the question was, it looked like, we are analyzing on the order of tens of thousands of samples worth of data. So the question is, if you had much more data, like say billions of people that had been sequenced, would that enable better science? And the answer is yes, absolutely. So like to date, some of the largest projects that people have done where they've done RNA sequencing have reached to the level of about tens of thousands of people. And even that is a huge, huge, huge leap from where we were just a few years ago in terms of how many we can, reasonably do, even in a large distributed international project. But that number, they keep rolling back that number. So like, for example, this year we've probably analyzed on the order of 60,000 or so samples, and 10,000 of those are all just from one big project called the GTEx project. I don't even remember what it stands for, Genotype Tissue Expression. But so we're just now at the point where a single project can reasonably sequence about 10,000 people's worth of this particular kind of data, RNA sequencing data. And absolutely, people are trying to push that envelope all the time. There's bigger and bigger projects that are being funded, and it's all being enabled by these leaps in sequencing technology that have reduced the price so much over the years. So yeah, absolutely, it'll only get better.
Okay, I think we're out of time. I'm sorry. We can take any additional questions. We'll be just outside. And thank you all for coming, and thank the speakers.